Okay, so thanks everybody who came here, and thanks to Omics. Thanks to Omics who invited me for this presentation. The field of my presentation today is the successful Leishmania organism, which is stirring immunity cells and mediators to achieve a trade-off balance useful both to host and parasite. If you think a little bit, this strange interaction is in some way the continuation of the One Health very interesting discussion that we have with my previous intervention, and is some more specialist for parasitologists. And it would be interesting to see how this protozoa, this flagellated digenetic parasitic protozoa, can lead to this very complicated equilibrium between the host and the parasite. Just to introduce Leishmania very quickly, Leishmania are parasitic disease caused by flagellated digenetic parasitic protozoans, including the order Kinetoplastidae, and are divided in two subgenera, Leishmania and Viania. Over 20 species infect mammals and may cause human and animal leishmaniasis. Animal leishmaniasis is a deadly disease. A lot of dogs and other canine are dying every year of this terrible disease. They are transmitted by sandflies that are smaller than other kind of diptera, and they are transmitted from the warm blood animals, including humans. This parasite needs two different kind of host, a vertebrate host and an invertebrate host. This is quickly the life cycle of Leishmania. And you can see on the left side, the red arrow indicate the stage that Leishmania can carry over the invertebrate. In this case, the insect, the sandfly. And the other side, the blue arrow, indicate the cycle that Leishmania can carry over the mammals, including human. Since my presentation is basically dealing with the cutaneous and tegumentary leishmaniasis, I introduce here four species of sandfly, the Phlebotomus papatasi, Sargenti, Perniciosus, and Perfilievi, which are supposed to make a transmission of this leishmaniasis. This is a poor insect, just taking a blood feed on human skin. And these are the 22 species of the genus Leishmania, which is supposed to have the organism who support different kind of leishmaniasis. So leishmaniasis conventionally are divided in three main groups. On the left side, you can see a form of cutaneous leishmaniasis. B, you can see a picture of mucosal leishmaniasis. This is a laryngeal infection, very strange, unusual, but both our group and other group in Italy observed a few of these strange mucosal infection. And the right side, you can see typical infection of the disseminated visceral leishmaniasis, which can be lethal if not cured. This is a conventional tables where all leishmania are divided based on geographical criteria and on kind and type of disease. You can see the Old World species, the New World species, and the kind of disease. At the very right end, you can see the cutaneous form, and then the mucocutaneous form, and then visceral form. This is just a map that I very, very difficult to find on web. Is an official map by WHO just 2 years ago about the epidemiology of cutaneous leishmaniasis. You can see both the New World and the Old World are red, means there is a very high incidence of cutaneous leishmaniasis, including Italy. You can see here Italy, where our study are carried over, and you can see is red, so high rate of cutaneous leishmaniasis. Sorry. This is the equivalent map, but regarding visceral leishmaniasis. Still just 2 years ago, so very up-to-date map. You can see here Italy is a reddish, let's say pink. So there is, of course, leishmaniasis, but is not so frequent. But the most frequent cases of visceral leishmaniasis are certainly in Brazil, in Sudan, and in India. In these 3 countries, there is a very, very large and deadly cases of visceral leishmaniasis. Okay, this just is a typical lesion. Stay there. This is a typical lesion of cutaneous leishmaniasis. You can see here before therapy is a kind of volcano lesions, and this is before therapy. After glucanthine, which is meglumine antimoniate, injected locally, you can see 1 month after therapy, the lesion is much improved. This is a film, but I think the film stopped, that Leishmania promastigote craving to penetrate into mononuclear cell. These long organisms are Leishmania, and they try to enter this cell that is supposed to be a mononuclear cell where they are breeding inside. Once penetrated into host cell, the promastigote quickly differentiate in amastigote. You can see here the amastigote like small violet particles, both inside in the picture A and outside in the picture B of mononuclear cell. It's just a reminder of the development of the T cell. Can you see the naive T cell can develop into the Th1, Th2, Th17, which is very up to date, and the Treg that we work a lot starting about 7 years ago, and other kind of T cell. But this is the real program of T cell when they encounter the promastigote of Leishmania. This encounter is very, very interesting by a biological and by a medical and veterinary interesting feature. Why? Because the Th0 lymphocyte start to develop and progress through the Th1 cascade or through the Th2 cascade. Th1 cascade is much better for the host because lately this leads to parasite elimination because the interferon gamma, TNF alpha, and IL-2 are some cytokine that help macrophage to increase the production of nitric oxide, can you see here, and other mediators that are directly lethal for Leishmania organism. So they kill Leishmania. But there is another deadly way, the deadly way of Th2. This way usually lead to B cell, plasma cells, and these plasma cells and other mechanism bring to the parasite dissemination. Why? Because the Leishmania amastigote inside the macrophage and different kind of monocyte cell still keep going, still going to breeding and multiply, and never macrophage is able to kill. And the Treg cell is the other important lineage. This lineage is producing IL-10 and TH-beta. These two cytokines here are very, very well known as inhibitory cytokine. They regulate in negative the cascade and bring to the limitation of the eradication of Leishmania. The focus of our studies during the latest years is leading by a question. Is Leishmania able to cause disease and escape host defenses in humans by increasing regulatory inhibitory immune mechanism? Sorry. Our contribution to understanding the mechanism of Leishmania disease include both in vitro model, where live protozoa are infecting human peripheral blood mononuclear cell, or another model, we keep going with dosage of serum inflammatory mediators as well as serology and immunology markers in Leishmania infected humans. Materials, I'll go very quick on this, just to say that protozoa strains are provided by the kind Dr. Gradoni and Dr. Gramicia from the Institute in Rome, and some purification were carried out to obtain the metacyclic promastigote, which is very, very interesting for the infection. In vitro infection of human PMC with metacyclic promastigotes and evaluation of mediator release at 4 hours and 24 hours after infection. And then, in some experiment, we evaluate the nitric oxide just in vitro by the Grease reagent. The other models include the cutaneous microscopy and the mucocutaneous biopsy specimen obtained from suspected tegumentary leishmaniasis. Molecular biology real-time PCR and of other aliquots of the same samples were carried out, and serology and immunology of serum samples with the execution of ELISA to evaluate IgG anti-Leishmania and to evaluate the C-reactive protein as a general specific inflammatory marker, and to evaluate also, we try to evaluate lymphocyte population, subpopulation, and the cytokine level. This is a very long story. I try to make as short as possible, just to indicate that in PBMC infection by Leishmania major and Leishmania infantum, we can see that there is a negative correlation observed between parasite concentration and the levels of TNF-alpha. In other words, when we use high level of Leishmania, we see that TNF-alpha, instead of increase, they decrease, okay, the amount of TNF-alpha. And when we use infantum, which is the most dangerous species because it is both dermotropic but is also viscerotropic, means that you can have with infantum both cutaneous, mucocutaneous, but you can have the deadly leishmaniasis, visceral leishmaniasis, and people can die. So with this, that is very interesting species, we can have even more this effect. So the more you increase the Leishmania, and the less is the TNF-alpha that you can see on the supernatant. And then you can see also the IL-6 was significantly increased by Leishmania major at 4 hours. On the contrary, the other species tested was unable to increase this IL-6. At 24 hours, both species caused a significant elevation of IL-6, but again, an inverse correlation linked the burden parasite, I mean the charge of the parasite, to the cytokine concentration. Again, very rapidly, MCP1 was the chemokine which is specific for mononuclear cell. So it's the same, is the equivalent of IL-8, but IL-8 is for polymorph nuclear cell, but MCP1 is chemotaxis, yes, but just for mononuclear cell. So MCP1, of course, was increased. And was increased why? Because the parasite need more and more mononuclear cell for breeding reason. So inside that cell that can be arrived after the MCP1 signals, more and more parasite will be multiplied inside the cell. So it's interest of this parasite to collect, to attract the large number of monocyte, macrophage, histiocyte, dendritic cell. Inside this cell, the Leishmania will multiply. So there's this very interesting observation. And the last but not the least, the nitrate analysis was carried out in order to estimate the release of nitric oxide, which is a stable nitrite metabolite. At 4 hours, nitrite levels was slightly but significantly elevated in comparison to medium-treated controls, following all concentration of the new species of Leishmania used. At 24 hours, very interesting scenario, because the lower parasite burden significantly enhanced the nitrite release, so the nitric oxide, after stimulation. On the contrary, the higher progmastigote concentration of Leishmania major and Leishmania infantum did not cause any significant change on nitrate level. It's the same story that TNF-alpha. You more give Leishmania and less have the mediator, Th1 mediator, which is supposed to prosecute and to stop and to inhibit the parasite. On the other side, you want to evaluate directly the mediator, I mean nitric oxide. You see that the more protozoa and the more parasite you bring inside the reaction, and the less is the nitric oxide. So something is wrong for the host, and something is good for the parasite. So this is the graph is exactly what I tell you before. So we go very quick. And this is the in vitro effect of promastigote, Leishmania major, Leishmania infantum on human PBMC cell. You can see here the very tall column for Leishmania, and this is the control with LPS. We need a positive control for the release of MCP1. You can see LPS, yes, significantly increase MCP1, but the increase of the Leishmania, the stimulation is much more the LPS. Why? LPS say, well, it's good, every cell is good, I don't need monocyte, I don't need that, okay? Every cell is good. But monocyte need to, Leishmania need the monocyte for breeding and multiplication. So for the more MCP1, the more monocyte, the more breeding. Okay, this is just the diagnostic that we carried out on our samples, I mean clinical sample, human samples. You can see here the picture of the specimen with conventional Giemsa stained slide. You can see the small corpuscles, violet corpuscles, usually inside macrophage. At the other hand, you can see here the PCR real time for Leishmania. I don't know, okay, I can see you very well. This is the Leishmania from for the humans, and these are the different color. I can't see that. Sorry for the color is very fading. And this is the color for different species of Leishmania. And so you can see the overlapping curve that identify as a melting curve the exact species of Leishmania. And then we try to repeat in this in vivo model the results defining the evaluation that we already did on in vitro model. And again, when we check for TNF-alpha level in 3 groups of individuals, you can see control group means is healthy volunteers. You can see here Leishmania negative group means patients but who are negative for Leishmania. And then you can see the Leish group that are positive for Leishmania by conventional or by molecular methods. You can see here the white column means the negative one, and the other, the Leish negative one, are very tall. I mean, there's not a lot of TNF-alpha, but they're tall. But on the contrary, Leishmania TNF-alpha column is very, very low. Why? Because again, Leishmania prohibit TNF-alpha. Why? Because TNF-alpha is the mediator that latterly will bring to killing of Leishmania. So Leishmania must inhibit it. And again, interferon gamma, we wish to have the same results with interferon gamma, but probably with interferon gamma, the mechanisms are much, much more intriguing and complicated. So we did not have this straightforward effect that we already had with TNF-alpha. Let's see the other one. IL-4 give very, very nice and expected results because IL-4, you remember from the previous slide, is the mediators which lead to Th2 cascade, which is the cascade which is preferred by Leishmania because bring to the inhibition of eradication of Leishmania inside the cell. So IL-4 represented the Leishmania positive IL-4 is this column is very tall, so there's a lot of IL-4 and the other control is very low. The same for IL-10. IL-10, which is the inhibitory cytokine, when you have the positive Leishmania, both with conventional test and molecular test, you have a very tall IL-10 column, and the control is very low. In this case, you can see MCP1 and IL-8. IL-8, it doesn't matter. I don't need that. I don't need neutrophils. Somebody say, neutrophil is the Trojan horse for Leishmania, and it's something you will enter, and that's it. Our results say something different. But anyway, the very important story, what is MCP1, because MCP1, you can see here, with the positive sample is very tall. Unfortunately, we have a tall column also for Leishmania negative, but Leishmania positive is even taller. In this case, you can see some interesting data about growth factor. Some growth factor, especially VEGF, behave itself the same way that TNF-alpha. Look like a pro-inflammatory mediators, which need to be prohibited, to be inhibited, to allow the progress of Leishmania infection. Okay, this very quick are some data about the single patients, the single positive patients, where we can find in microscopic examination of the mucous or cutaneous samples, the amastigotes of Leishmania, and most of these are carried over also the molecular test, and we can see Leishmania again. The interesting point of this table, which is long to discuss, but we limit the discussion at this. Usually, people say that cutaneous Leishmania is negative to IgG, which means several things. Probably one day is not enough to discuss this point. But we discuss just the clinical diagnostic, no more. Okay? We show here that approximately an half, probably more than 1/2 of our patients with tegumentary leishmaniasis are instead positive to the IgG, which means a lot of things. Means, first one, the infection is a long story. because otherwise you cannot have IgG positive. Second, the infection is an organismic infection. It does not kill the cell or attack the cell in the small piece of skin where you can see the volcanolation. But actually, in the long story, probably months, probably years, attack all the organism. And so the immune system is aware. Some Leishmania is somewhere, in some temple, somewhere. Nobody knows where, but some place there is Leishmania. So, first conclusion, you should check for IgG anti-Leishmania, even in a tegumental relation, okay? Because sometimes, no, not always, of course, but sometimes you can check positive value of IgG anti-Leishmania first. Second, the Leishmania is a chronic infection, and there's a lot of time for the immune system to be aware that some Leishmania is somewhere. And third point, we can see, okay, this is the IgG. We check above the total, which is increased. The third column is always the Leishmania positive group. Again, here is increase, is a IgG1, is increase in Leishmania positive group. We did not found some significant results with IgG4. And this is one of the latest graph is dealing with the subpopulation of lymphocyte. You can see the CD4 is a little increase. CD8 is completely the same for positive and negative group. And this is the most interesting for us picture, because this column, which is very, very high, very significant, is the column of the CD25 with Leishmania infection. This means this group is the group of infected patients where CD25, which is the lineage, which produces a lot of IL-10, so the lineage which commands the inhibition of the immune system is very, very high, okay, in these infected patients. Conclusion. So we go back to the first contribution. Our contribution to human Leishmania infection suggests that first, the parasite causes a depression, a strong depression of Th1 immune response in favor of the Th2 and Treg immune cell lineage and their cytokines. The in vitro model of Leishmania infection that I showed before, as well as the evaluation of markers or mediators in clinical samples of Leishmania patients, underline the same above reported feature. The in vitro model of Leishmania infection was associated to the inhibition of nitric oxide, the main host mediator, which direct Leishmania killing effect. The presence of parasite, or after some time, years, or maybe months, years of disease history, evaluated by conventional molecular methods, is a bona fide proof of long-lasting Leishmania infection and strongly support the lack of a rapid eradication of parasite. If you find the DNA of Leishmania or its kinetoplast, or you can find the amastigote of Leishmania in your lesion, Leishmania is there, it's still there. Interestingly, almost half of parasite infected patients showed positive anti-IgG, the IgG anti-Leishmania in serum samples, even in the tegumentary infection, where you do not expect that, thus demonstrating the long-lasting immunity against the parasite. The last slide, all these things together should support the idea of a trade-off between the parasite and the host, beneficial to both of them. Why? Because Leishmania strain is able to survive and disseminate within the host, I mean different tissue, different organs of the same host, or out of it, because some sandfly can have blood feeding and transport the parasite to another host. But on the other hand, the host is gaining a long-lasting immunity, long-lasting attention against a potentially lethal pathogen. Because let's say that I have my stupid cutaneous lesion of infantum or another organism of the Leishmania group. Then I go for hobby, for work, for job to India, where there is Leishmania donovani all over. Okay, some fly, okay, have a blood feed on me and want to transmit Leishmania donovani. That is potentially deadly. But my immune system, both the cellular, as you can see here with the lymphocyte that you check every lineage, and the immune cell of the other lineage are aware. And so the immunity can act rapidly to eliminate or to halt the parasite. So that's the way the succession of the 100 or 1,000 years, these two species, the humans and Leishmania, grow together. Thanks for attention.